If you're not sure what is your final patient population, for example, what is your annual demand, if there is uncertainty here, or if you run a multi-product enterprise, then it may be smart to have a small and flexible facility and not overload on capex investment, which you have when you get a large stainless steel six-pack plant. And so that uncertainty, that is certainly a characteristic of our time, I would say, that would cause me to favor a small scale facility. And so perfusion is one of the strategies where you can use relatively small bioreactors to generate a large volumetric productivity.
Hey, smart scientists! Welcome to another episode of the Smart Biotech Scientist podcast. Are you dealing with perfusion processes, or you're wondering whether you should develop a perfusion process? Well, you are in for a treat today, because I'm having a conversation with Jochen Sieck, who is the Director of Cell Culture Media and Process Development at Merck Life Science. And we are going to be talking about perfusion and sustainability, which is a complex equation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Jochen, to the Smart Biotech Scientist podcast.
Hey, David. It's a pleasure to be here.
Absolutely. It's really a pleasure to have you on the show today. Jochen, share something that you believe about bioprocess development that most people disagree with.
Well, I would probably go for media cost or media price has a relatively low impact on process economics. And that's based on media performance can get you much more product and then dilutes your cost base. And so it's much more impactful to have a well-performing medium than a cheap one.
All right. Now, before we dive into our topic about perfusion today, let's talk about yourself for a moment. So how did you get started in biotech, and what pivotal moments and decisions brought you to your current role?
Well, I'm a biotechnologist by training and started in the biopharma industry as an intern at Novartis in bioprocess development, 2008. Then was later hired in the same group as a PhD student, later a postdoc. So then I joined Merck, became a lab head, first leadership role in 2014. Also later studied an MBA, and recently I was appointed global head of media and process development R&D in Merck Life Science. A pivotal moment was probably, I would have almost stopped PhD actually midway. So there was a reorg going on in Novartis, and my supervisor at my university had, lost interest in the project. And so I needed to find two new supervisors, both for the company and in the university. And so this is where I will be eternally grateful that Professor Morbidelli from ETH stepped in and adopted me as his foster child PhD.
Life isn't always linear, isn't it? And it's so great to have the right people.
Oh, true. This whole career sounds like there's a thread going through, but it looks like that retrospectively. But if you've been in there, then it's really a different story.
Absolutely. I could totally relate to that. My career has not been linear either.
I know, it's very impressive.
And I think what I'm also hearing in your career is it also depends on the right people that go this journey with you.
Absolutely. I consider myself very lucky to have met some of the people that have been starting the whole field of mammalian cell culture in bioprocessing, some of the people that have been doing the early work with Joe, the first chemically defined media, getting rid of the serum. So I was able to learn from some of the people that have really started this whole industry, including the first chemically defined media developers. So I had great mentors over my career. That's definitely an important thing.
And since you have a great overview over the whole industry, I would be curious, Jochen, what do you think are the key challenges that biotech scientists and engineers often face in bioprocess development?
I will relate mostly to mammalian cell culture, right? Because this is my area of expertise, and it's a very peculiar area within the biotech field, I would say. And that is because you have a lot of unknowns. So I believe this is one of the reasons why parts of the biopharma industry are so conservative, because that's the only strategy from a scientific point of view to manage all those variables and all that uncertainty. So, for example, we have a growing understanding of what's going on inside a cell during a bioprocess. But this is really a complex field. And once you have solved it for one cell line, you can open the next project to talk about the next cell line. So it's a complex story. Then cell culture media, of course, my home topic. There is between 50 and 100 chemicals involved in there that could all chemically interact. So there is, again, a growing and large understanding of what can happen, what can go wrong, but it's certainly not a solved problem. Then if you think about a bioreactor, so you have multiple surfaces that interact with each other. You have a variety of interactions between the process and the cell and the media, which are all interlinked. So that is really adding to a lot of complexity. So once I made a joke to some of my fellow PhD students at ETH that were chemical engineers and working on downstream processing and they constantly teased us, the biotechnologists, of, you guys have no idea what you're doing. I said, in biopharma, in mammalian cell culture, you have to be comfortable to juggle black boxes. So you have a couple of systems that you don't have sufficient insight, but you can control the inputs and outputs, like in a black box model. But you have multiple of these, so you need to juggle. And I think that's a good picture to describe bioprocess development.
I like that, Jochen. We are juggling black boxes. That's a great way. Probably next time I have a conversation with my people who are not engineers and scientists, I just tell them, hey, I'm juggling black boxes.
Yeah, from a theoretical point of view, that's a fair description. It's not always satisfactory, but it also brings home the point that you should try to minimize the impact of the uncertainty. And so here we talk about chemically defined media versus the olden days where you would add sera or lots of hydrolysates and stuff that is not characterized, for example.
So let's dive into another, let's call it black box, or our main topic of today, perfusion and sustainability, a complex equation, because perfusion processes have been around for many decades. Different commercial molecules such as factor VIII were launched using perfusion processes. And then the industry moved to fed batch processes. And recently again, the shift from the traditional batch or fed batch went back to continuous and integrated manufacturing. So what are some of the recent developments in perfusion, and why actually are we moving back to perfusion?
Maybe before going into the future or the current stage, I'd like to comment taking one step back on why people were using perfusion. You mentioned factor VIII, of course. That's the traditional and ongoing need for perfusion to work if you have a non-stable product in the end of your bioprocess. So I think this is also something where perfusion still has its application today. The second historical reason to go perfusion back in the days of spin filters and the like was that the protein yields of the cells were so low, so that using perfusion was basically the only possibility to get to a sufficient volumetric productivity of your bioreactor process, to get sufficient drug to treat a number of patients because the yields were so low with the cell culture in these days. That has been overcome. As I said, so this journey has been taken several decades, but it's been very impressive how the yields in fed batch processes have increased over the years with the adoption of more robust and stable cell lines and higher performing media and processes. So I think that was the main reason fed batch became much more popular, because it's simpler to operate and the yields were increasing to that point. Now, as the whole industry has more or less been locked into fed batch, it's difficult to think about change again. But there are some important business drivers that, from my perspective, favor perfusion or continuous processes. And this is, if you're not sure what is your final patient population, for example, what is your annual demand, if there is uncertainty here, or if you run a multi-product enterprise, then it may be smart to have a small and flexible facility and not overload on capex investment, which you have when you get a large stainless steel six-pack plant. And so that uncertainty, that is certainly a characteristic of our time, I would say, that would cause me to favor a small-scale facility. And so perfusion is one of the strategies where you can use relatively small bioreactors to generate a large volumetric productivity.
So what I'm hearing, Jochen, is that back in the days, the drivers were mainly scientific or technical drivers because the products were not stable and the productivity was so low. Whereas today in the Perfusion 2.0, let's call it like that, the main driver is more a economical or strategy driver to minimize cost and minimize risk.
Yes. So if you build a new facility today as a biopharma company, independent of scale and size, you are well advised to build a flexible facility, as in contrast to considering the blockbuster facilities for the monoclonal antibodies in the past.
And besides the economical drivers, are there any other advantages of going back into perfusion with a cell line that is a hutch? Well, I say it a bit more in a controversial way, obviously.
I was waiting for years to make that pun again. We are going to go back to the future because today's perfusion is not the same as we would be going back towards 10, 20 years ago. So for sure, we have different tools today, different cell retention devices, more powerful media, better understanding, better cell lines. So it's not going back. It's, if you want to excuse the pun, it's back to the future. So there's more aspects to this as well. So you mentioned the sustainability aspect here. And also there, I think it's more or less similar picture. So building a new stainless steel facility, by the way, stainless steel facilities, due to the, for example, energy demand for autoclaving, are in itself a bit problematic from an energy consumption point of view. So on the other hand, we have plastic use in a more single-use based facility, but it's a different order of magnitude by all I know. And so that is also one of the reasons why small and more flexible facilities may be a smarter investment than continuing with this large stainless steel cathedral paradigm.
And if we talk about perfusion, well, you mentioned already the sustainability aspect. So one thing that comes into my mind is obviously the media consumption, because if we have a perfusion process, even though the bioreactors are much smaller, the media consumption is high. So how do we navigate that tension?
So one of the key issues or drawbacks of perfusion is exactly that. It is the media logistics. So if you look at it from a cost point of view, and there are some good cost models that are available through the Bioforum, for example, comparing fed batch and perfusion facilities and single-use and stainless steel facilities, you can see that the cost structure, if you operate perfusion, is of course a bit different. You get a higher amount of variable cost associated with operating perfusion due to the media that you have to provide. On the other hand, again, the volumetric productivity is really the big advantage of operating perfusion. So from the same, for example, 2,000 liter bioreactor, you can only produce so many fed batch lots a year, but you can get that same amount of productivity as in a much larger vessel if you run that 2K bioreactor in perfusion. So ultimately, it comes out how many grams or kilograms of biopharmaceutical product you can produce using the same amount of inputs, for example. And if you make that equation, then I think it's the same drivers ultimately that drive the economic optimization of processes and the sustainability optimization of processes. You want to do more with less. It's a generic engineering goal, to do more with less. And it's the same paradigm that you need to follow to become more sustainable. Do more with less.
Yeah, I like that. That's a great way of seeing it as engineers and scientists, do more with less. Before we look into some practical strategies to use less media, I was just wondering about another scenario, because there are some molecules that require very specific quality attributes, for instance, biosimilars. There are also some other scenarios. I'm just wondering whether that could also be a driver to go into a perfusion process instead of using a fed batch.
So I have a limited experience here, to be honest. For example, produce a biosimilar in perfusion where the originator was produced in fed batch. But what I can share is that, of course, the process has also an influence on the product quality attributes. But then again, back to the future, I think one of the issues with the whole biosimilar field is that you're trying to mimic process performance that is really bad, because it's like from the process technologies that we had available, from the cell lines, media, and process parameters that were state of the art 20 years ago. Before you went into clinical trials, you had to define that process. So some of those drugs on the market today, they are really based on old technology. And then with the biosimilar, it's sometimes the issue that you have to be as bad as in the past if you look at the quality attribute. So in general, I think if you have originator that is produced in perfusion, it's much smarter to make it in perfusion today as well. And then if you want to swap from a legacy fed batch product and make that biosimilar in perfusion, that is adding on the complexity, I'm afraid, because you have most likely a change in cell line. You could use modern cell lines. You are using more modern media. That is all giving you potentially different quality attributes. And so also changing the process can, if you're lucky, it can undo some of the changes that you have to adopt due to using a new cell or media. But if you're unlucky, then it may actually deter the product quality attributes further from the originator. Then you have to look very closely on what are really the critical quality attributes and why were they defined the way they are. And we have quality by design paradigm by the health authorities, for example, that would probably encourage you to look at. So what are the real drivers that drive patient outcomes? And so I'm actually in favor of the bio-better strategy, although I'm not 100% sure how successful that strategy is with regards to health authority feedback. But I like the idea that you want to improve the process as you try to target the same pharmaceutical goal. Instead of going back in time 20 years ago and try to make a protein that was originally manufactured using, from today's point of view, overly old and bad performing tools, if you would redo those processes today, you would probably get a much better product. And so I would favor that.
So this means, Jochen, that perfusion can really make sense for a new biological entity because you can control quality much better. Since you're operating in, let me say, so-called steady state, probably not the biological, but let's assume it's a steady state. Whereas for a biosimilar, it's very challenging because you enter so many new variables.
Well, that's of course very right. So with perfusion, you get a relatively constant quality output. That's definitely a given. So even if you don't consider it a steady state by the hard chemical engineering criteria, I would say you can get sufficiently stable temporary steady state that gives you a very robust quality. While in a fed batch, you get an accumulated quality profile that includes the quality made during the growth phase, during the static phase, and during the decline phase. Plus you get chemical degradation depending on the residence time in the bioreactor. So a variety of fates for each of the proteins. So depending on how long they have been in the bioreactor. That is different, of course, in perfusion. I was more referring to if you can actually control the steady state to a level that would provide the same CQAs as a fed batch, and given what I just said. So that's probably challenging.
Now, let's look into strategies, how we can make perfusion more sustainable. Jochen, can you give us a few steps or strategies how to lower the perfusion rates?
So there's multiple angles to that. Again, if you follow the paradigm, do more with less. I mean, the goal for the optimization from a sustainability point of view needs to be very similar to what you would normally consider perfusion medium optimization. Is you need to provide the optimum environment for the cells to proliferate sufficiently, but not too much, and to produce the product of interest. And you want to strip the medium of any components that are not serving this purpose, not serving this goal, but you need to provide the cell with all the nutrients that it requires. I am to this day continuously amazed that although we are all using CHO, it sounds like we are all doing the same thing, how sensitive CHO cells can be to small changes in nutrient quantity. Sometimes you can get large jumps in performance up or down, depending on what you're looking at. So this is really the whole trick. Similar for perfusion as for other processes, you need to provide the cells with the exact right amounts of the right nutrients.
Yeah, this is definitely key to guarantee optimal cell growth and optimal, obviously, quality and optimal yield and at the end of the day. Now, what are the critical components, and are there some specific considerations that our listeners should keep in mind when they are optimizing the cell growth and the productivity?
Well, it's very cell line dependent, obviously. In general, what you can say is that while in fed batch, you have a relatively elegant possibility to re-enter nutrients as feeds, where you can use a whole box of chemical tricks to increase the nutrient concentration. So you can play with pH, you can use certain components, you can switch certain components so that they are more soluble or more stable, for example. So that is a luxury that you do not normally have when you talk about perfusion, because it all needs to be in the medium itself. So that's one of the main challenges I would see. So typically the feeds contain very high amounts of amino acids, and so we need to get them into the perfusion basal medium. And so that's, I would say, one of the key challenges.
Hey, smart biotech scientists. I hope you've gotten as much value out of our conversation with Jochen Sieck as I have. Jochen Sieck is the Director of Cell Culture Media and Process Development at Merck Life Science. And in part 2 of our conversation, we are continuing our journey about perfusion and sustainability, which is a complex equation. And we are going to look at strategies to reduce the media consumption in perfusion processes. So stay tuned, and I'll see you in part 2 of our conversation. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.